Hey guys, this is going to be our DNA replication video, a topic I totally just forgot about. I should have done DNA replication actually with you just before we did operons, but switch those two lessons, no big deal. So you should remember DNA replication from freshman year, that basically it's this process of, I'm going to draw a sort of a twisted up double helix here. The double helix untwists into its straight ladder shape, right? So there's our first step. And then it unzips, right? That's the hydrogen bonds breaking into two halves of the molecule. And then new nucleotides that are present in the cell base pair with the open strands, forming two new sides, and we have two full molecules now, right? So this is like the freshman year version of DNA replication. Basically, 3 steps. The double helix untwists, the double helix unzips, and extra nucleotides present in the cell base pair with the open strands, right? So obviously in AP Bio, we have to step up the complexity a little bit. Now, before we do, I just want to point out to you that when you have a newly formed DNA molecule, you have 1/2 old strand and 1/2 new strand. Biologists refer to this as DNA replication is semi-conservative. Right? And you have a activity to do, or an exercise, I should say, that replicates the experiment that confirmed this. Okay? This is the Messelson and Stahl experiment. And I just want to walk you through this, apply the concept real quick. What Messelson and Stahl did was they wanted to find out if DNA replication was conservative, which would basically mean from a parent strand, you produce that same strand, and you produce a totally new strand. Right? That would be conservative DNA replication, and it does not happen that way. DNA replication could potentially be dispersive. Not a word you need to remember necessarily, but dispersive DNA replication would look like this. It's kind of hard for me to draw it real quick, but it would just be, there's just like old and new throughout, something like this. I'm not doing all the pictures, but there's a picture on the doc of this. I can actually just finish it, right? Like if it was just completely mixed throughout, that would be dispersive DNA replication. Those are not the case. The DNA replication is semi-conservative. So how do we know this? Well, what Messelson and Stahl did was pretty ingenious. They took DNA. They were getting DNA from bacteria. So that's a piece of bacterial DNA right there. And they culture the bacteria in a medium, in a solution that contains nitrogen. So the bacteria is going to incorporate the nitrogen into the DNA as they replicate DNA, because obviously you need a source of nitrogen to copy DNA, nitrogen bases and all that stuff, right? So they incubated the bacteria in a medium with nitrogen 15, which is a heavy isotope of nitrogen. And then they transferred the bacteria to a solution containing nitrogen 14, which is a lighter isotope of nitrogen, and then they weighed the molecules, right? And so if you just sort of do this as a thought experiment with me, if all the black here represents nitrogen 15, right? And if I'll use pink, if that represents nitrogen 14, right? And we've transferred the bacteria to the new medium, so their existing DNA is going to be made of the heavy, right? So we're gonna replicate the DNA. We're going to separate the double helix, and when the new strands fill in, they will be with the heavier element like this. So you want to ask yourself here at generation 2, what percentage of the DNA molecules have the lighter element? You could also pretend, by the way, you could also do this experiment with a radioactive isotope, right? Where you could measure radioactivity. So what percentage here would have radioactivity, or what percentage here would incorporate the new nitrogen? And the answer, of course, is 100%, right? Both the molecules have that element in it. Now let's replicate the DNA one more time. Here's my old nitrogen, right? Here is the open strand. All I've done in this first picture is I've just separated the two molecules, right? And now I'm going to fill in all the new strands with my lighter element. Or you could think of it as your radioactive. I'm drawing some very sloppy double helixes, but you get the idea. Now what percentage of the DNA molecules contain some old and some new? Well, only 2 out of the 4 are like that, right? And so this is only 50% of them contain the old strands. If you do this one more time on your own, you can sort of measure how the percentages go. And that's how Messelson and Stahl were able to determine, okay, we know that DNA replication is not conservative and not dispersive, but instead it's semi-conservative.